from the University of Göttingen. Oh, something happened.
It was the recording. It's okay.
Oh, all right. Yes. So I'm pleased to introduce Marc Appelhans from the University of Göttingen. He is a systematic biologist, researcher, botanist, who has done much work in family Rutaceae. He has done extensive molecular research as well as field work. I have heard illustrious stories of him working with June Wen in the field and Papua New Guinea and places in Southeast Asia. I think at one point it involved cutting down a toxicodendron species. And I think all he was eating for many months was broccoli and rice. But those are just some of the tales that get better with age, as the years go by, these stories, they tend to grow. But hopefully the stories you will hear tonight about this family and its systematic relationships will be something to really enjoy, especially since it's Christmas time and we're enjoying all things citrus. There is no Christmas without oranges and candy canes, and there is no January Botanical Society of Washington meeting without a citrus family Rutaceae phylogeny. So with no further ado, I give it to you, Marc.
Thanks, Gabe. Yeah. Thanks for reminding me of the broccoli and rice diet I had in New Guinea. I almost forgot that. Yes, yeah, nice memories with June Wen in Papua New Guinea. Okay, so yeah, also from my side, Happy New Year and welcome. For me, it's almost, it's 1 a.m. in the morning, but I'll try to do my best and stay awake. But I had a nap before, so everything's going to be fine. So the title of my presentation is Phylogenomic Studies in Rutaceae, the citrus family. I think the first thing you all have in mind is, of course, lemons and oranges, so the genus Citrus, but it's just one of many, many genera in this family. And I'll try to cover the diversity of the family during this presentation. Actually, Washington, D.C. is quite a nice area or quite a nice city for a presentation about citrus because of this gentleman, Walter Swingle. Yeah, he was a citrus taxonomist. He also worked on cultivars, hybrids, also on citrus diseases. And he worked at USDA for an incredible 50 years. But I think he was always going back and forth from the DC area to Florida. But you might have heard this name, and also one of the Rutaceae genera is named after him. Okay, so the structure of my talk, I will briefly introduce the family Rutaceae, show some pictures of morphological characters and so on. Then I will talk briefly about the two methods that I'm using. They're called HypeSeq and RedSeq, and they are two of these next generation sequencing methods that are very popular right now. And then the biggest part of the presentation will be about a new family level phylogeny that I'm working on based on HypeSeq using the so-called Angiosperms 353 kit. I will talk about that later on. And I arranged the slides in a way that I will just go through all these different clades in this phylogeny, show you pictures of the plants, tell a little bit about them, show you the diversity. And at 2 points in the phylogeny, when it's about the genera Xanthoxylum and Melicope, I will zoom in and talk about 2 projects that I'm working on regarding these 2 genera. But the majority of the talk will be about the family level phylogeny. Okay, starting with the introduction. So Rutaceae are, it's quite a big family that we find in all tropical and subtropical areas worldwide. You see the distribution map here. It's rarely found in temperate zones with just a handful of species always. And it's particularly species rich in Southern Africa and also in Australasia. It's quite a big family with about 150 genera and about 2,000 species. And only 2 of the genera have more than 200 species, and those are Melicope and Xanthoxylum, that are the ones that I mentioned already. Yeah, we have important crop plants in the family, of course, like all the different citrus species and hybrids, but also Xanthoxylum is quite important in especially Asia, but also Japan, Korea, and other Asian countries. It's the source of the Sichuan pepper. Here's some morphological characters. So Rutaceae are mainly trees and shrubs, so woody plants. Leaves can be simple or compound. All these leaf characters occur in the family. The most important spot character to spot a Rutaceae in the field are these glandular dots, the so-called secretory cavities that you see in the leaves and also in other parts of the plants. So they are usually present. Sometimes they are small, but usually they are visible if you hold a leaf against the light. And what's also very important is what's in these cavities. Yeah, the secondary compounds, essential oils, limonoids, alkaloids, many different compounds, and very characteristic for the family. The flowers are mainly relatively small and not very showy, and the fruits are quite different. So you all, of course, know the citrus fruits, which are called Hesperidia. So the Hesperidium is a modified berry, but berries are actually quite rare in the family. So what you mainly have are the fruits that you see on the left-hand side, also like all these small pictures here. So dehiscent fruits, capsules or follicles, and sometimes the seed remains attached to the open fruit, and sometimes they are expelled from the fruit. So this is the most important fruit character in the family, or the most abundant fruit character. There's one more picture I want to show. So what's also characteristic is this nectary disc between stamens and carpels. This is also something that I would say almost all Rutaceae have. Yeah, but otherwise they are quite diverse in their characters. Since I'm talking to people from North America, from the States, I would like to show at least one slide of the diversity in the United States. So only the native species, we have about 80 species and 9 genera. But if you look further down, of these 80 species, 55 belong to the genus Melicopi, and they are all endemic to Hawaii. And also 4 Xanthoxylum species are endemic to Hawaii. So about 60 of the 80 species that you have in the United States are endemic to the Hawaiian Islands. So only 20 species in the mainland United States. And I think the only 2 that you have in the Maryland, Virginia, DC area are Xanthoxylum americanum, the common prickly ash, and Teleatri foliata, the hop tree. Yeah, everything else is rather in the south, basically Florida and California have some species. Okay, yeah, so this is what I wanted to talk about in the introduction about the family very briefly. I would like to talk a little bit about the methodology I'm using. So this is all about next generation sequencing. So these new sequencing technologies that target, I don't know, maybe the whole genome or at least big parts of it. So the first method I'm using quite often is called HypeSeq. And in the literature, you find also the terms target enrichment and hybrid capture. So this is basically all the same thing. So what's done here? You target a certain number of genes, usually a couple of hundred genes, and you have to separate them in some way from the rest of the genome. And you do that with so-called baits. The baits are RNA molecules, and they hybridize, of course, to the certain gene areas in the genome. And these RNA molecules have a biotin tag. And biotin is a molecule that binds to another molecule called streptavidin, and there are magnetic beads that are coated with streptavidin, and if you add them to this to your mix, you can apply a magnetic field, and then all the target genes with this biotin tag are dragged to the side, and you can wash everything else away. Yeah, and in this way you can separate your DNA of interest from the rest. Then you would amplify it, and then you would sequence it, in my case, using Illumina technology, so one of these common next-generation sequencing sequences. The question here is, of course, which genes do you select? Yeah, you need genes that are informative for your taxon, so you need mutations and so on. In these genes, you don't want to have any housekeeping genes that might be completely conserved and don't give you any information. But you also don't want genes that are too variable. So maybe if it's too variable, you get into alignment problems. You cannot really make proper gene alignments. So you need information, but not too much information. And of course, you also need genes that are present in all, or at least in most of your species that you're interested in. So this is how you select your genes. And then there is one possibility to skip all that. because there are these pre-designed kits. One of them is the so-called angiosperms 353 kit. This is a set of baits for 353 genes that occur throughout angiosperms and are informative. And I'm using this pre-designed kit for the family level phylogeny. But if you are interested in a single genus or something, really something more specialized, it might be better to create or to custom make your own bait set. And this is what we did for the Xanthoxylum study, which is based on one genus. If you want to make a custom-made bait set, of course, you need to know which genes do you want to use. So you need some genomic background. So you need some transcriptomes or some genomes so that you can really make a selection and see which genes are suited for your study. Yeah, so those are the two strategies, custom-made or pre-designed kits. And I will talk about both of them later on. So the second methodology I'm often using is called RADseq. That stands for Restriction Site Associated DNA Sequencing. And here you make a DNA extraction and then you treat your DNA with restriction enzymes. Restriction enzymes have specified cutting sites, so they always cut a DNA strand at a certain position. And these cutting sites are the starting points for your sequencing. So you add for Illumina sequencing, you always need adapters. So you ligate the adapters to the cutting sites, and then you get homologous regions across all your different species that you're sequencing. Cutting sites, that's a little bit of a problem because with increased evolutionary distance, you get more and more mutations in two different taxa. And it can, of course, be that a mutation is directly at a cutting site. You see that here in the figure at individual 2. You see here in the middle, this black bar is not separated into 2. So here the cutting did not work. And that means you don't get this bit of DNA here. So you have some missing data here. And this is getting more and more important the more separate or the bigger the evolutionary distance between your taxa is. But basically you cut it with a restriction enzyme, and the starting points of the sequencing are the cutting sites, and that's why you get homologous sequences. Okay, that's like in one more slide, I would like to compare the data sets that you get from these two different methodologies. In HypeSeq, you have a couple of hundred genes, let's say 300 to 500. And in the end, your alignment will be, the gene alignment will be about 100 or 200,000 base pairs. RADseq is much, much bigger. You get tens of thousands of like small homologous regions, but they are like maybe 100 or 150 base pairs long. But still, this all sums up to a data set that has more than 1 million, maybe up to 3-4 million base pairs. So much bigger. In both methods, you can multiplex samples in one Illumina run. So one Illumina run costs about $1,000, and it's of course cost effective if you can multiplex several or many samples. If you look at the size of the HypeSeq data set, it's much smaller, so you can multiplex many more samples than with RADseq. An important thing for my studies is that HypeSeq works generally well with herbarium specimens. I'm working on a genus like the prickly ashes and Xanthoxylum that occurs in about 100 different countries. I cannot go to all of them, so I'm really happy that I can work with herbarium specimens. RADseq doesn't really work well with it. I think we tried a couple of times. Sometimes it worked, sometimes not. But HypeSeq, that's definitely a plus for the method. HypeSeq also often yields many plastid sequences. So sometimes you get the whole plastome, so the whole chloroplast genome as a bycatch for free. So this is another second source of information next to the nuclear genes that you are targeting. And then HypeSeq is more variable when it comes to taxonomic levels you work with. You can use this angiosperms bait set if you work on a family level or something. If you work on something like a genus, you can make your own bait set that is specifically designed for that study. RADseq is more limited to population genomics or to studies of closely related taxa. The problem is the mutations in the cutting site, so you get a lot of missing data the more you go down or on an evolutionary timescale. Okay, so these are the two methodologies. I just wanted to mention them. I cannot go into too much detail, but I think you are rather interested in the to see some plants and to see the diversity than like a very dry methodology-based presentation. So this was all I have about the methodology. So now we can zoom in to the diversity. So as I said before, I've worked on a family level phylogeny. 2 years ago, or in 2021, I published a Sanger sequencing project that contained 135 genera of Rutaceae, which is about 88%, which was really a very big step up because previous family level phylogenies had between 23 and 38% of the genera. So this was a nice, let's say, wrap up of the Sanger sequencing period. And now I'm involved in a project that is housed at the Kew Gardens in London. It's called the PAFTOL project. It stands for Plants and Fungal Trees of Life. And here they are using throughout angiosperms this angiosperm 353 kit. And I'm analyzing the Rutaceae data set in the framework of this project. And here we sampled 125 genera, so about 81% of the diversity, but there is some more information from this GAP project, Genomics for Australian Plants, coming in. And just before Christmas, I went to Leiden to the Netherlands to collect more herbarium specimens. So this number of this 81% at genus diversity will increase in the future. So you see on the right-hand side of the screen, you see a phylogenetic tree, and this is what I will now go through clade by clade. I will show you what kind of genera we find there, how they are distributed, what their morphological characters are. But one general thing, so we now divide Rutaceae into 6 subfamilies, and we have also a dated phylogeny that tells us the origin is quite old. It's in the Cretaceous at about 100 million years ago. Okay, so let's go through these different clades now. So yeah, here we have an overview of these 6 subfamilies. You see that they are not really of the same size. You see the first one has more than 100 genera and 1,700 species. It's the majority of the family, and the others are rather small. So it's a little bit unbalanced. So the circumscription of these subfamilies is relatively new. For a long time, we took the publication by Engler, which is about 100 to 120 years old, and Engler subdivided Rutaceae in, I think, 6 subfamilies, and he paid a lot of attention to fruit characters. And I plotted the fruit characters on this tree that you see at the bottom, and you can really see it's kind of like a mix of colors. So especially Engler divided one subfamily or designated one subfamily to genera with drupes, and then he said they are completely different from the ones with the dehiscent fruits, so with capsules or follicles. But we see many sister group relationships in the phylogeny of drupaceous and dehiscent fruited genera. So this really doesn't hold up. So the only fruit character that's important is the berry and Hesperidia type fruits for the citrus relatives. Everything else is not suited to define subfamilies. Okay, so now let's go through these different subfamilies. I start with the one that's at the bottom here, the Neoroidae. Basically so I did my PhD about this species poor and quite old subfamily. It was always one clade, but now with this PAFTOL data, we see them as 2 independent lineages here. So they might not be monophyletic. But here, so in this world map, you see the distribution, so mainly tropical, but there's one species also in the Mediterranean region in Europe. And I can show you some line drawings of these genera, and then you will see they look very, very different. Yeah, so for example, Harrisonia is a very nasty spiny shrub. Then Neorum is maybe half a meter high from the Mediterranean and Canary Islands with simple leaves, and Trimeris and Tetrameris flowers. Bottigoa has these winged fruits. Cedrolopsis and Pteroxylon have winged seeds. Dictyoloma also has winged seeds, but in a different kind of way. So there the wing goes once around. And then Spartelia and Sonreia look completely different. They actually look like palms. Yeah, so they are monocarps. They have unbranched up to 10 or 15 meter high stems with big compound leaves. And yeah, they're monocarps, so they produce these huge inflorescences and then they die. Yeah, I have some pictures. So those are 2 of the 3 species from Jamaica. And Spatilia glabrescens, I think, grows up to 15 meters. And the specimen you see there is at least 10 meters tall. So they are very odd for Rutaceae. Yeah, so they have this palm-like appearance. And there is one species of not Spatilia, but the sister genus Sonreia in Brazil that makes inflorescences that are up to 3 times 3 meters. They are really huge. Yeah, and then the plant is 20 meters tall. And after flowering and fruiting, it dies. Yeah, so these are 2 species from Jamaica. I have another 2 species from Cuba. So this is me collecting Spatilia splendens in Cuba. This is, yeah, splendens means pretty or precious. And this is a very pretty species. It has, I think, more than 100 leaflets per leaf. So very, very spectacular, very nice, good looking plant. But this Spatilia splendens, morphologically, it's different from the others by the number of leaflets. But if you look in the phylogeny, I don't know, for example, here on this side, you see splendens here and here. So it's probably not monophyletic. So I think that especially in the Cuban species, we have a lot of hybridization going on. So the Sanger sequencing studies show that most of the species are not monophyletic. So I would like to study these using RedSeq. So I'm now talking to a friend from Cuba, and we want to write a grant proposal together. So this is quite a particular genus. In this subfamily Neuroideae, I wanted to show you one other study that I did using or based on the genus Neorum. Actually, this genus, it looks completely different from Spatilia, of course. It only has 2 species, one from the Mediterranean region, one from the Canary Islands. You see both of them in the pictures. But a 3rd species has been described from Cuba about 120 years, I think, ago. And since then, Neorum has been used as a model organism to explain like disjunct distributions by breaking up of Gondwana and by plate tectonics. And it was always believed that the genus is very old. But actually, this 3rd species from Cuba doesn't exist at all. So the only one from Cuba that I've seen that is a Neorum is the holotype, which is actually in my university now in Göttingen. So that's the image on the left-hand side. This is actually a Neorum, but it's morphologically and also genetically not distinct from the Mediterranean Neorum tricoccon. So the holotype is about 150 years old. And I managed to get DNA and managed to get DNA sequences from it. So I was really happy about this. And so it's really not distinct from the Mediterranean species. And everything else I've seen in other herbaria that is labeled Neorum trimerum is actually something else. Yeah, it's either Hypericum or Scaptia. So none of the labeled specimens in herbaria are Neorums. So my conclusion is that it's probably a misplaced herbarium specimen. It's one of the specimens collected by Charles Wright. And all our Charles Wright specimens from Cuba have proper labels, handwritten labels. And this is the only one that doesn't have a label. So probably it just fell down. And there were like on the bench, there was a pile of specimens from Cuba and another one from Italy or something. And someone threw it on the floor and just, well, okay, let's put it on the Italy pile. Yeah, so I think this is what happened. There is no Cuban Neorum, and it's not such an old genus, and the distribution has nothing to do with plate tectonics and the breakup of Gondwana land. Okay, so those were the 2 stories about the Neuroideae. So I'd like to move on to the next subfamily, the Rutoideae, which are actually quite weird for Rutaceae as well, because most of them are herbs. Yeah, Rutaceae are mainly woody plants, but except for the early branching here, for Chloroxylon, everything else is herbaceous. Yeah, I think you might know Ruta graveolens maybe as an ornamental plant from the United States. Thamnosma occurs in the United States. The other ones are Asian. So they are herbaceous plants. I would like to talk about 2 of them. Xylopaganum is a very rare and monotypic genus from China, and it's more or less endemic to the area of the Three Gorges reservoir area where they like dammed the Yangtze River, and it's more or less extinct from the wild. Yeah, it's like all the native distribution areas have been flooded. So it's very rare. I was really happy to sample it in the phylogeny from specimen grown at South China Botanical Garden. So that's that. And the other one is Thamnosma that occurs in the United States. Yeah, you see here in this map a distribution. So you have it in like California to Texas and then down to Mexico. But that's just part of its distribution. So we find other species in Southern Africa and then other species in Eastern Africa, in Yemen and Somalia. Which is a very weird disjunct distribution. But if you look at the seeds, it has these protuberances. So I really think it's sticking very well to birds' feathers. And in this way, it's eventually managed to get to all these different places. Yeah, and Thamnosma is definitely a monophyletic genus. So it has been studied about 10 years ago. It's a monophyletic genus. Okay, so like I mentioned the Rutaceae subfamily before, so there are some morphological characters for this group. They have a herbaceous habit, except for this one genus, Chloroxylon. Usually the leaves are compound or at least deeply lobed, except for Thamnosma. And then there's one character that they all have. They have an increased number of ovules per locule. I think it's between 6 and 10. Usually Rutaceae have 1 or 2. So this is really a good character for this subfamily. Okay, so that was it about the Rutaceae. The next two subfamilies, I would like to talk about them in one slide because they are quite small and I don't have so much to say about them. So one is called the Haplophyloidae, consisting only of the genus Haplophyllum. Haplophyllum used to be a subgenus of Ruta in the past, but now we know it's not closely related to Ruta at all, and now it's even in its own subfamily. And then, so Haplophyllum is an old world genus, like Mediterranean, and I think it goes like to Kazakhstan, maybe Western China. And the other genera in this sister subfamily, the Amyridoidae, they are neotropical. And I can only say something about Neoridium and Amyris, which both occur in the United States. They look quite different, but they have both fleshy fruits, and then they have one character that's very characteristic. They have only one carpel, which is also very rare for Rutaceae. So they have one shared character despite their overall dissimilarity. Okay, so now I'd like to move on to the next subfamily, the Auranthioidae, so the citrus relatives. I think that's the subfamily that most of you know best. So this is the part of the phylogeny here. I mentioned in these blue boxes genera from Africa. So actually, we always connect Auranthioidae in citrus with Asia. But actually, in these three clades, we also have African taxa. So we have 3 independent colonizations to Africa. Down here, Citropsis, and then these 3 genera, they are all endemic to Africa and some to Madagascar. And then Clausina up here is actually a Southeast Asian genus, and one species also occurs in Eastern Asia, in Eastern Africa. Yeah, but in general, for the Auranthioidae, the genus boundaries are often not very clear. So citrus is, of course, very well studied, the rest not at all. So there are really phylogenies missing for most of these genera. And there's also a high percentage of monotypic genera, which is also quite interesting. And yeah, I would like to now in the next years go through most of these genera and publish phylogenies for these. Okay, so that's for citrus, of course, we think about the edible fruits and citrus in particular. I will talk about that, but also Walter Swingle, whom I mentioned in the beginning, made a tribal and subtribal classification. So in the way I organized these bars here, it looks like everything is nicely monophyletic, but actually many of these things are not clades, but grades, like the tribe Clausinae consists of, can you see my mouse? I think so, right? Yeah, okay, consists of this clade and then this clade. So it's actually a grade, not a clade, but the tribe Citraeae is a monophyletic group. And then the subtribes are actually, some of them are also grades, not clades. And there's one subtribe that is not sampled here, Micromelinae, which might be the sister group to all Auranthioidae because it's the only one with valvate petals. Everything else has imbricate petals. So this is also something that is missing in this tree. Yeah, but then the subtribes within Citrinae. So I'm just moving one slide further. So Walter Swingle also gave English names to these subtribes. There we have the Balsamocitrinae, the hard-shelled citroid fruit trees, and they are actually not monophyletic. So we have this here and this and this. So 3 different clades that all have a hard-shelled fruit, but actually they are quite different. So this subtribe is not monophyletic. Okay, so but now let's talk about the different edible plants in this subfamily. You are all aware of the different citruses, so I don't have to mention all of them here. I just want to show you some other pictures of things that you can eat. You might know the bale fruit from India and other Southeast Asian countries. The limeberry triphasia, I think it's also occurs, or it's invasive in some Caribbean islands, maybe also in Florida or also Texas or something. It might be invasive. So these you can also eat. The curry tree, Bergera koenigii. Here you can eat the fruits, but usually the leaves are most interesting. So the Indian curries all have the leaves, not the fruits in them. Yeah, so there's more than just citrus that is edible. But like if you look at these, they don't all look like citrus fruits. So there are different kind of fruit categories in this group. So let's say the early branching clades have typical berries. They might sometimes be a bit dry or leathery, but otherwise they are typical berries. And then you have these hard-shelled fruits in 3 different clades, and as said before, they're quite different. So this bale fruit group and these African genera here, they have hard-shelled fruits filled with resin-like gum, and then the elephant apple Limonia has a very specific condition. It has in flower, the carpels have 4 to 7 locules, but it all fuses into one single locule when the fruit develops. So very different. And then Swinglea, the tabok tree from the Philippines, has also very different fruit types. So it's not really hard, it's not woody, but it's a leathery fruit, and it has mucilage glands, not this resin-like things. And then everything that comes down from here, like everything in this clades, is the typical Hesperidium, so the berry with the pulp vesicles that we know from citrus, yeah? Where these pulp vesicles grow into the locules. Okay, so different kind of fruit types, but they are all derived from berries. Okay, so now I kind of skipped citrus for now. I just want to show some slides based on citrus. I haven't really worked on citrus, so I'm showing slides that David Mabberley gave to me some years ago. And I just want to show here what is a species and what is a hybrid. And I think these are 15 slides. I will just go through them very quickly. So if you start from the mandarin and the pomelo, if you cross them, you get to oranges and grapefruit. And then if you add a third botanical species to the mix, the citron, if you cross that with the oranges, you get to the lemons. And now it's building up and becoming more and more complex. So yeah, it's very, very complicated, and I haven't really worked with this genus, but actually this next generation sequencing area gives now very, very deep insights into citrus and the native and the botanical species and the hybrids. Yeah, but basically citrus, yeah, no one really knew how many species there are. So Mabberley and also Walter Swingle said there are about 25 to 30 species, but the estimates go from 6 species in the Engler treatment about 100 years ago to 167 species by Tanaka. But yeah, now we more or less know it's about 25 to 30 species, but there are some former genera that are now included in citrus. I guess you all know the kumquat fortunella or the trifoliate orange Poncerus. They now all belong to citrus. But everything that we buy, like all the commercial citrus cultivars and hybrids, they are basically derived from 3 important paternal species. You know, the citron, the mandarin, and the pomelo. Most of the other things are hybrids. Because they are so important economically, we have genomes sequenced. Yeah, so these 3 parental species have been sequenced and some more, but not all of them have been properly annotated. But you really see there is some research being done based on next generation sequencing. Okay, so this is what I wanted to talk about regarding the Auranthioidae subfamily. So far I talked already about 5 out of the 6 subfamilies, but only about roughly 20% of the species. So now we still have this huge subfamily Xanthoxyloidae in front of us. So this is, 80% of the species. And within this, so I will go through this now subclade by subclade. And I would also like to talk about 2 case studies regarding the genera Melicopia and Xanthoxylum. So I will just always do that when I'm at this particular subclade, then I will zoom into these 2 genera. Okay, so the first of these subclades of the Xanthoxyloidae is kind of like I would call it the trash bin of the Rutaceae. So they are genera that look completely different and from morphology would never come to the idea to unite them in one clade. Actually, the first one, Stauranthus, I cannot say much about it. I've never seen it. It's a neotropical genus. But then Casimiroa, you might know, it's called the white sapote or sapote in English. It's an edible apple-sized fruit. Also Central and South American, and I think it also occurs in southern, I think in southern Texas. They have Casimiroa. So these are the neotropical ones, and then we have Dictynus albus. It's a monotypic genus from Central Europe. So actually, this is the only native Rutaceae in Germany. And it goes all the way to Siberia and Kazakhstan. So it has a very wide distribution. And it's actually quite pretty and also very untypical for Rutaceae. So it has these zygomorphic flowers, and quite showy and big flowers. And it's a herb. Yeah? So it's quite untypical. You would expect to find it more often as an ornamental plant in gardens, but it has really aggressive compounds. I think they are called furanocoumarines. When you are exposed to the sunlight and you get these compounds on your skin, you get really a very nasty rash. So that's why they are not very popular as ornamentals. And then you have 2 more, Skimmia and Oryxa. They are both Eastern Asian. Oryxa is endemic to Japan. Skimmia, I think you all know the Skimmia as ornamental plants. And then Oryxa, again, something that looks completely different. Oryxa has a very nice phyllotaxis, so the leaf position. It always has 2 leaves to the left, 2 to the right, 2 to the left, to the right. You see it here, like 2 here. Then you have 2 on this side, 2 on this side, 2 on this side. It's very, very particular. You really cannot miss this. I think it's the only plant in angiosperms that has this leaf position. Yeah, so they look completely different morphologically. They have this very large distribution, and they are all very species poor. I think Skimmia and Casimiroa have a couple of species, that the others are monotypic. Yeah, so this is a weird clade. So then let's move to the next one. And this is now a very nice geographically and morphologically circumscribed clade. It also has a subtribal name, Diosme. Here we have the first genus here, Calodendron. It's a tree, and this is quite widely distributed in southern Africa. Everything else is confined to the Cape fynbos vegetation in South Africa, so a very narrow distribution range. But it's a quite species-rich group, so almost 300 species, but half of them belong to the genus Agathosma. We only sampled 1 here in this phylogeny. But from earlier studies, it's known that Agathosma is not monophyletic. And except for Calodendron, they are all rather small, like shrubs or subshrubs. They have all simple leaves. They have no endosperm, which is kind of untypical for Rutaceae. Like the citrus relatives also have it, but everything else has usually has endosperm. And they have these horn fruits, which is also quite characteristic for this group. Yeah, but then also, of course, the distribution is quite narrow and quite clear. Okay, so then we have the next big clade in this Xanthoxyl. Oh, and yeah, some images. So yeah, you see really they are quite small and they have these simple leaves. There's one interesting genus, the one that you see on the lower part here, Implurum. It has very, very small and not really showy flowers, and it's the only Rutaceae that I know that is wind pollinated. Everything else is classically insect pollinated, some bird pollinated plants, but this one is wind pollinated. Okay, so then we have a larger American clade over here. And I will divide this into 3 subclades and talk about them. So the first one, quite genus and species poor. It's also one that is not really, that doesn't really have any apparent morphological similarities. I think you all know the hop tree, Telea trifoliata. It's part of this clade. Everything else, is also of course neotropical or American. There's one genus that's missing that's also quite popular as an ornamental plant, Choesia. We know from the Sanger phylogenies that it belongs to this clade as well. But you see, like from the images down there, they are quite different, and there's no apparent morphological character for this group. The one that's quite off is Platadenia. So this the one with these compound leaves here. It was believed in the past that it's rather related to the prickly ash, to Xanthoxylum, but it belongs to this Telea choesia clade. Okay, so this is also a clade that really doesn't have any good morphological similarities. The next one, so the last part of this American, I still have one more to go. So again, one with only 4 genera here. This is also a very small clade, Central and South American. I don't know these so well. So Pilocarpus is quite a beautiful thing with these long and hanging inflorescences. And then 2 other genera in this clade, Helietta and Balfurodendron. Helietta again occurs in the United States, I think with 1 species in Texas. And they both have also winged fruits. Yeah, this is what we saw before in Telea in the hop tree. And in the past, these 3 genera, so Telea, Helietta, and Balfurodendron, were united in one subtribe, the Teleinae. But yeah, you see now from this presentation that these 3 genera are not closely relatives, and actually Helietta and Balfurodendron, the fruits look quite different from the hop tree, I have to say. Yeah. Okay, so these are the 2 American clades that are relatively genus and species poor, and now we come to the 3rd American clade that is quite species rich, and we have here again a Central and South American plus Caribbean clade, quite species rich, about 175 species. And this is a clade that has very good morphological characters. I don't know this clade so much. If you ever would like to hear more about it, I suggest to invite Jackie Kolunki from New York. She is a specialist on this clade. She retired, I think, 1 or 2 years ago, but maybe she would be happy to give a talk. So these have very good morphological characters. They have zygomorphic flowers, which is very rare in Rutaceae. They have their petals basally joined. Usually Rutaceae have 3 petals, and they have staminodes and pseudostaminodes. I think the pseudostaminodes are like outgrowths from the petals or something. That's why pseudostaminodes. But it's something that most of these genera have. And yeah, so at least 1 or 2 of these characters is joined by all of these genera and all the species in there. Again, like in the citrus relatives, we have a high percentage of genera that are monotypic. So 8 of these genera are monotypic. Yeah, so they deserve some more work. Okay, so this is all I have to say about this clade. Then I will zoom into the next one, and this is actually the first clade where one of my case studies is placed. So Xanthoxylum is part of this next clade. It's a clade that has an informal name called Proto-Rutaceae. So in the 1970s and 80s, the secondary compounds of this group have been studied, and so these 5 genera, these 4 genera have very, very similar compounds, and they have been considered primitive. So they always thought they would be like the early branching clade in Rutaceae. That's why they call them Proto-Rutaceae. But you see now it's actually deeply nested within the family. Philodendron, the cork tree, Tetradium, and Phagoropsis are quite small. Yeah, so I think Tetradium has 9 species, the others just 2 or 3. But Xanthoxylum is quite a species-rich genus with about 225 species. And as I said in the introduction, it's commercially important as the source of the Sichuan pepper. And we also have, I think last year, a genome of 1 species was published. So I would like to talk a little bit more about Xanthoxylum. And actually, it's part of an ongoing DFG project. DFG is the German NSF, and one of my PhD students or grad students is working on this genus. So it's the only Rutaceae genus with a pantropical distribution. I mentioned already more than 200 species, and I mentioned the use as Sichuan pepper. I think the most characteristic feature is these prickles and knob thorns that you see on the branches, also on the leaf racquets, also on the leaf blades of some species. So it's quite easy to spot in the wild. The flowers it's a dioecious genus. And it has two different flower morphs. So let's say the tropical and subtropical species have normal flowers with calyx and corolla, as you see at the bottom image here of this Hawaiian species. You have calyx and corolla. The more temperate species have very reduced and small flowers that only have tepals, so not differentiated in calyx and corolla. So this is a quite interesting diversity of flowers. And then also Xanthoxylum has polyploids with one species up to 144 chromosomes. And we have apomicts, so species that reproduce asexually via seeds. That's why I became interested in this genus. And we are now in the process of creating a hype-seq phylogeny of this genus with a custom-made bait set. I have to apologize for the image here. My PhD student is currently traveling, and she only gave me this PDF of the tree with annotations in red, so I cannot really zoom in here. But I think the individual species names don't really matter so much. So we have about 350 genes in this data set, about 250,000 base pairs of aligned data sets, so quite a big data set. We have many samples and species included, and we have about 144 that are currently being sequenced. That will be added to this phylogeny in the next weeks. We use the pipeline called Hype Phylomaker, just to be complete. I mentioned this name here. And then I will immediately go to the results. So the current sectional classification, which is based on morphology, is problematic, but I would rather focus on some other questions. For example, what's the geographical origin of this genus? It's pantropical, so what's the origin? What are the major dispersal routes? So what about these two different flower morphs? Have these reduced flowers in the temperate species originated only once or several times? And what about apomixis and polyploidy in this genus? Have they one origin, or did it originate multiple times? Okay, so first the origin. We have an origin definitely in the Old World. So everything that you see in blue is African, and in dark blue is a clade from Madagascar and the Mascarene Islands. So this is the first, the earliest branching clade that we have here. The second clade that you see here in red is the species Xanthoxylum asiaticum. It used to be placed in a separate genus in the past. And despite the name, it's distributed even more widely than Asia. So we find it throughout Asia, really from China to all the way down, or actually all the way down to Australia, and then also in Africa and Madagascar. And it's apparently all the same species, and we now sampled specimens from throughout this distribution range, and it's really a monophyletic group, and they all look very much the same. So it's really, let's say a good species and a monophyletic clade. Okay, so this is also occurring in Africa despite the name. So the origin is definitely in the Old World, and this is also supported by fossils that we find, for example, also in Europe, where we don't find any species today. Now then the next clades are basically the Asian species with calyx and corolla. This is the light blue box here. And then we find a clade from Australia, New Guinea, the Philippines, and Pacific Islands. And here, again, Hawaii is very interesting here. So the Hawaiian clade, which consists of 4 species, is quite separate from the other Pacific species. So we have 2 independent dispersal events to the Pacific, one to Hawaii, and the other one rather this stepping stone dispersal from New Caledonia to Fiji to Tahiti and to the Austral Islands in the South Pacific. Yeah, so the Hawaiian clade is not nested within the other Pacific species. Okay, another thing that's interesting about the Hawaiian species, chromosome counts are available only for one species, and this species, Xanthoxylum hawaiiense, is octaploid with 144 chromosomes. The other species there, it's unknown. I hope I can get material so that I can count these chromosomes there. But yeah, octaploid, what does it mean? It could be that it's a hybrid, an allopolyploid, yeah, where two species hybridized that resulted in a ploidy shift. This is what could have happened here, because we also, based on the HypeSeq results, we made an analysis called quartet sampling. Quartet sampling is a method that analyzes phylogenetic conflicts or incongruence, and we have a very strong signal at the base of the Hawaiian clade that really points to a hybridization before Hawaii was colonized. Yeah, and because this one species is octaploid, the hypothesis is that this hybridization resulted in a ploidy shift prior to the colonization of the Hawaiian Islands. Okay, so this is about the Hawaiian species here. We have some more clades to go. What you see here in this blue box is two South and Central American clades, and then we have in this red box in both of the figures, we have species that are American and Asian. Yeah, so if we concentrate on this left figure here, we have Xanthoxylum americanum, the common prickly ash, Xanthoxylum clava herculis, and then other species from the southern United States and also from like Central America and Mexico and the Caribbean islands. And then we have, as a sister to this, the temperate Asian species. So that means, and all of these in this red box have these reduced flowers. And we see here that it's actually a grade, it's not really a clade. And there are other species from the Caribbean and Central America that are closer to this temperate Asian and clava herculis clade than to Xanthoxylum americanum. So the question is, did this reduced flower evolve 2 times? Yeah, or was it lost here again, or was it reversed here again in this other group here? The second question is the ones with the reduced flowers. Some of them have been described to be apomicts, but it's not really studied for all of them. So the question remains, are all of these apomicts? And this is something that we want to study in the next 1.5 years. And then one other thing is, because we have temperate Asian species nested here within this whole American clades, we have a dispersal event from let's say temperate America to temperate Eastern Asia. So that means first Xanthoxylum jumped over to America, and now within this temperate clade here, it jumped back to temperate Asia. Okay, so this is basically the phylogenetic tree. I would like to sum up this a little bit. So we have for Xanthoxylum an Old World origin. We have 2 independent dispersal events to the Pacific based from Asia. Actually, there's a 3rd dispersal event from South America to the Juan Fernandez Islands that belong to Chile. I didn't talk about that, but 2 independent dispersal events from the Asian region to the Pacific. We have a dispersal from the Old World origin to the Americas and back to Eastern Asia. So it once moved around the world. We have at least 2 clades with octaploid species. I mentioned Hawaii. The other one is within these reduced flowers. So one of these temperate Asian species also is octaploid. And these tepaloid flowers, these reduced flowers, evolved either once or twice. The topology is not really clear there. Okay, so I talked a little bit more about apomixis in the introduction. And as I said, this is an ongoing project. We want to do some more chromosome counts to be able to determine the ploidy level. There's another a method called flow cytometry, where you can measure the size of the nucleus, which is also an indicator of the ploidy. And we would also like to do some microscopic analysis of the embryo sac development. Because in Xanthoxylum or in other Rutaceae such as citrus, the embryo often develops not from the fertilized egg cell, but from nucellus cells. And this is quite characteristic for apomictic plants. And if this is the case, often more than one embryo develop. Yeah, so more than one nucellus cell develops into an embryo, and then we get this polyembryony that is shown here for citrus. One seed gives rise to 2 or 3 different plants. Okay, so this is what we do. So like the methods sound a little bit old-fashioned, but I think like microscopy and chromosome counts, it always gives very important insights in addition to this molecular studies. Okay, so that was it about Xanthoxylum. So let's move on in our Xanthoxyloidae tree. So the next slides will be quite briefly. So this is now a clade that I could have separated into 2. So there's one group here, the genera Vapris and Ivodia. They are African, and basically everything else is Australian. Yeah, so there we have one of these intercontinental connections, but here these Australian genera, they are actually quite different. So if you look at this picture of Flinders with these weird spiny capsules, it's quite different from other Rutaceae, and like most of these genera here, they are species poor and they have some odd morphological characters. And there are 2 genera missing here in this clade, the genera Pitavia and Acradinia. They belong to this clade. I know this from the Sanger phylogenies. And these are actually very interesting. So they look very similar, you see from these images here. But the interesting part is the distribution. Acradinia occurs in southern Australia, including Tasmania. Pitavia is endemic to Chile. And so here we have one more of these southern continent disjunct distributions. And based on morphology, Tom Hartley from Australia, he always said, oh, these 2, they look so remarkably similar, but he didn't have any proof from a molecular phylogenetic study, but now we have this proof. So they are really close relatives. They're actually sister genera. Okay, so this is very quickly. The next clade I want to mention, I also do this in one single slide. This is also nearly endemic to Australia, plus some neighboring islands. And these are several genera, 14 genera, quite many species that mainly occur in the dry areas in Australia. So dry open vegetation, most of them are actually smaller shrubs or subshrubs. They all have 5 Maris flowers, and they have alternate leaves. So there's good morphological evidence again for these. There are 2 cases where the flowers look quite different. Korea, which is also an ornamental plant, has again fused petals. Now this is a bit withering here. And then this one, Diplolena, it looks almost like an Asteraceae flower head. So it has bracts out here, and these are the small flowers that are all clustered together. But apart from these 2, they all look quite similar. 5 Maris flowers, alternate leaves. So good morphological characters, plus the distribution in rather dry Australia. Then the sister clade and the last clade is actually also very common in Australia, but also in New Guinea and some other parts of Southeast Asia. But these are the Australian rainforest taxa, and they are all tetramaris or 4 Maris flowers, and the leaves are usually opposite. And so they are from the vegetation different, but also from the morphology. Yeah, so here we have a couple of genera, and the most important for my studies is Melicope, which is the largest genus of Rutaceae, and we find it from Madagascar all the way to Hawaii. So it also has a very wide distribution. Several genera are nested within this genus, and Warren and I have already started to make it monophyletic by merging some of these genera in it. But in this clade, there are also other species-rich genera, for example, Baronia with 150 species, also quite important as an ornamental plant. And others with like 50, 60 species. Yeah. So I would now, in the last minutes of this talk, zoom in and talk a little bit more about Melicope, and I will focus only on the Hawaiian radiation. In total, we have about 230 species, and we have this big wide distribution from Madagascar to far into the Pacific. The Pacific is not the result of one single dispersal event, but we have several dispersal events to the different island groups here. But I will only focus on Hawaii. On Hawaii, it's the second or third largest plant radiation. And all the images you see here on the right-hand side, they are all from Hawaii. And yeah, when Gabe invited me to give this presentation, he told me, "Oh, can you also give some pictures of field work, because people always like to see some more images about field work and collecting plants." So we went to Hawaii, I think 4 years or 5 years ago, and we went to the 4 biggest islands. And I just want to show you some pictures of the Hawaiian Islands, starting with Kauai, the northern or the northwestern one, and also the oldest island. So it has pretty much a very rugged landscape. Like always when Hollywood needs a tropical scene. So for example, the opening scene from Jurassic Park, I think they fly in here with a helicopter. So this is quite a very scenic and quite a popular view. This is the same thing viewed from the sea, yeah, from the boat. Kauai also has the only canyon on the Hawaiian Islands, the Waimea Canyon. Our field trip was often like very steep, so I don't know if you really see this, but the slope is actually quite steep here. We sampled a lot of specimens, but then also it has these swampy areas, these higher altitude bogs, which are very species rich. So this was a very productive day in the during our field trip. Oahu, also deep valleys here. Here we also often concentrated on the higher altitude regions. You might know this Diamond Head close to Waikiki Beach on Honolulu. We didn't go up there, but we went to the other mountain ridges on Kauai. We sampled from like more like a drier vegetation zones, but mainly from these higher altitude, very wet zones, which also have these steep cliffs, which you see on this picture, for example. And yeah, so very species rich, high altitude regions, and it was really a very nice time that we spent on Oahu as well. Okay, I couldn't, I just selected many different pictures, but I think from the mosses and so on, you see how wet these vegetations are there. Maui, we also focused mainly on the higher altitude areas, because this is where most of the Melicopi species are, also very, very wet and moist areas. And again, these swamp areas we visited. But then, of course, Maui is also the place where we find the Haleakala crater and the volcano. And this is kind of like a moon landscape. And here, for example, you can see the Hawaiian silver swords, which were nicely in flower when we were there. Then the big island, Hawaii, we spent only, I think, 3 days there. And we didn't collect so much there. But again, you see here one of these rainforest habitats. And then we did some sightseeing. So there were some minor lava flows here. And of course, as a tourist, you go there. And of course, you can also see the succession states there, where you see the first shrubs coming up. So that was also very interesting. Okay, so now you got some impressions from the Hawaiian Islands. I would like to spend the last couple of minutes talking about Hawaiian Melicopi. So we have some research questions here. So how many successful colonizations or events were there from, let's say, Southeast Asia to Hawaii? This is a question that has been studied for a long time. For example, the Hawaiian lobeliads. It was always believed that they derived from 5 or 6 independent colonizations, but actually it's just one clade. Yeah, so most of the genera that came to Hawaii colonized the islands only once and then moved from island to island there. What's the geographical origin of the group? Yeah, so where does the Hawaiian lineage come from? When did Melicopi colonize the islands? So in terms of millions of years, most of the Hawaiian flora and fauna, so like the iiwi, like the honey creeper birds, but also the silver swords, they arrived about 5 million years ago, which coincides with the age of Kauai, the oldest of the current main islands. So we wanted to see, is it the same for Melicopi? Then Hawaii is so isolated. So is Hawaii a dead end of dispersal for Melicopi, or did it escape? Or did it jump somewhere else based on Hawaiian ancestry? And then how does the island-to-island dispersal work? Yeah, so how did it jump from Kauai to all these other islands? And here yeah, this dead end of dispersal, I want to give you one example, the koa tree. It's believed that it also occurs on, I think, Mauritius, close to Madagascar, which is quite surprising, but it means that for some genera, Hawaii is not a dead end of dispersal. Okay, so so this inter-island or the island-to-island dispersal, I would like to show you on one slide how this often works on the Hawaiian island. We have the so-called progression rule, or speciation on a conveyor belt. Yeah, so it's jumping from the older island, let's say Kauai, to the next emerging one, to the next one, to the next. And when it colonizes a new emerging island, there's little competition, ecological niches are forming, so there can be a radiation, and then it jumps to the next youngest island. Back colonization to older islands happens as well, but it's less frequent because all the niches are occupied. And this is what we can actually see from a phylogeny. You can really see there's a clade from Kauai, one from Oahu, one from Molokai, one from Maui, one from the Big Island. Yeah, and if there has been a significant speciation, you don't see a single line here, but you see a clade or a grade from these islands. Yeah, and this is what happened also in Melicope. Yeah, but first let me show you this phylogeny. So the first lines here is just like technical things. So we studied it using the red sequencing method, so the one with these restriction enzymes, and it's a huge data set. So we have about 500,000 variable sites in our DNA alignment. The whole alignment is, I think, like 3 or 5 million base pairs, and 500,000 are informative characters. So a big data set. We sampled 81% of the Hawaiian species. Sounds okay, but not perfect. But actually, some of these species are believed to be extinct. Yeah, so you can never get to 100% using red sequencing. The phylogeny is well resolved. I showed these support values only for the main branches, but also like, yeah, I didn't have space here, but these branches are also nicely supported. But it shows us that the morphology-based classification is not natural. Like I showed in these color bars here, I show the different sections of Hawaiian Melicope, and actually only the Palea group is monophyletic. That's the green clade here. The others, like Apocarpa, has 2 clades, and then the sections Cubicarpa and Megacarpa are completely mixed up. So Palea is a monophyletic section. The other one we have to modify. And then we have here this Platydesma clade, which is actually was always believed to be an endemic genus to the Hawaiian Islands. It looks different from the other Melicopes because it's a bird pollinated thing. It has the anthers form this tube, and the leaves are also very different, and it has like this palmoid habit. But actually, it's deeply nested within Melicope, so it is a Melicope, and we already reduced it to Melicope, so the genus Platydesma officially doesn't exist anymore as a separate genus. Okay, so that's about the taxonomy here. We made a dated phylogeny, and the origin of this whole clade is about 8 million years. So it's significantly older than the island of Kauai, the oldest of the main islands. We also have a very high diversification rate in the past 1.5 million years, but I think this only tells us that along these branches there has been a lot of extinction, which of course we cannot sample anymore. In a molecular phylogeny. But going back to the origin, about 8 million years. And we think, so in this figure, you see this red line, which marks the age of the island of Kauai. And this is this is the age of Hawaiian Melicope as a total. We think that Kauai was colonized twice, and then from there the colonization went further to the other current main islands. But these 2 colonizations, of course, they did not come separately from Southeast Asia, but probably from nearby islands. Yeah, so we have this volcanic chain where the Hawaiian Islands are part of, and of course there are older islands. For example, Necker and Nihoa used to be quite big islands. Now they are quite shallow without a dense vegetation. But this might be the origin from where Melicope colonized Kauai 2 times. Yeah, so we have 2 colonizations, but actually just 1 colonization to the volcanic chain itself. Okay, and then within the islands, it largely follows this progression rule pattern. So if you focus, let's say, on this upper half of the tree, you have green, 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 green here, which stands for Kauaian samples and species. Then it becomes yellow for Oahu. Then it becomes red for Maui or Maui Nui. And then we have here one group that is like this lilac color, and that's the big island. And we have here one back colonization from Oahu, from Maui to Oahu. Yeah, so largely follows this progression rule pattern. But even more interesting is that Hawaii is not a dead end for dispersal. So we have two independent colonizations. You see them in the tree marked with these red arrows here. Two independent colonizations to the Marquesas Islands. So this figure is not up to scale. The Marquesas Islands are far, far more south in the South Pacific. And actually, Melicope is the only angiosperm genus where two independent colonizations out of Hawaii to the Marquesas Islands have been documented. Yeah, so this is really something special and not really expected, yeah, from such an isolated island group to another very much isolated island group. Okay, yeah, to sum up Melicope and also the whole thing, red sequencing is very well suited to resolve the phylogeny. As I said in the beginning, red sequencing is usually used for population genomics, but it also works very well for closely related species, such as a Hawaiian radiation. The morphology-based classification is problematic. Only one of the sections is monophyletic. This former genus Platydesma is actually a Melicope, and it has adaptations to bird pollination, and that's why the flowers look different. The lineage itself is older than the current main islands, so it has possibly been colonized twice from nearby islands such as Necker or Nihoa. The dispersal from or within the Hawaiian Islands usually follows the progression rule, and it's not a dead end of dispersal, so we have two independent colonization events from Hawaii to the Marquesas Islands. Okay, so this is what I was planning to talk about for Melicope, and you saw already in the family phylogeny, we are at the bottom of the tree. So that means I'm at the end of my presentation, and yeah, roughly 1 hour. This leads me to the last slide, the thank you slide. I would like to thank you all for listening, for coming. And I would like to thank Warren and June for being my postdoc supervisors 10 years ago. And luckily, we never stopped working together. I'm really happy about this. Then there are three more students that were involved in the Melicope and Xanthoxylum projects, Lee, Claudia, and Niklas. And then like the founding bodies, the DFG, the German NSF, and then particular the Kew Gardens and the GAP project for all these family level data that they all financed. And yeah, I'm happy to take questions now, and I would like to thank again for listening. And I think I will leave the slides open so we can just jump from slide to slide.
Oh, thank you so very much, Mark.
Yeah. Would it be better if I end the PowerPoint, or what would you say, Gabe?
I think this is fine, at least for the initial questions. Yeah, thank you so much. What time is it where you are now?
It's now 2 o'clock, 2:15 a.m.
Well, thank you very much for staying this far and giving such an invigorating talk at such a late hour.
You're welcome.
Throughout all your talk, I realize a lot of these plants grow in wet areas, but so many of them seem to grow in chaparral environments. Is fire an issue with its life cycle? Has there been much research that you can talk to about the nature of fire repression and or a need for fire burns for its growth?
Yeah, I think maybe in Flindersia, so that's the one with these very prickly capsules. I think I read that they need fire to germinate, but it's not very. It's not like some of the pine species in North America or like the Banksias in Australia. I think it's not such a big deal for these plants. Although I have to say, in the past, I used to buy a lot of seeds and I tried to grow them at home or in our botanical garden. And some of the Australian genera, there it was mentioned in the recipe how to grow them. It was they included smoke paper, so you were supposed to burn the paper. It produces a lot of smoke, and that would cause the seeds to germinate. So I think, at least for the Australian species, it might be an issue. Maybe not really like burning, like in some of the Banksias or in the pinus trees, but that at least they need some fume or some smoke that there has been a fire, so that they can germinate on these now exposed fresh land. So I think it's a topic, but it's not really much studied in Rutaceae.
Well, thank you. Yeah, I just figured with all the oil in the leaves, I didn't realize that all the genera had oil in their leaves.
Yeah, they all have.
Surely that would do something to ignite a fire.
Yeah, actually, the one that grows in Germany, Dictamnus albus, it's some people believe that it's the burning bush from the Bible. Yeah, because Dictamnus can like if it's really hot, like 40 degrees Celsius or something, the essential oils catch fire and it makes like one flame, and then it's then the fire stops again. But that's why it's believed to be the burning bush from the Bible, but it doesn't occur in Israel at all. But yeah, so they are inflammable.
Fascinating. Thank you.
Mark, great talk. What a whirlwind run through the whole family.
Yeah.
Fascinating and very accessible to all of us. I wonder a couple of things. One, on just a culinary note, are any other xanthoxylums like the Szechuan pepper, or is it just that one species that is used culinarily?
Is it the one? Yeah, no, it's several species. So there, like, I think it's a group of 3 or 4 species that are called Sichuan pepper. Then Nepal has, I think it's called the Timut pepper. Then Japan has what we call the Japanese pepper. So they're like Sichuan pepper is the most common name, but there are several species used.
And they all have the similar properties. And
yes. I'm not aware of any American species that are used as a spice, except that.
Fantastic stuff, though.
Yeah. So, but I know that, like, I think Xanthoxylum claviherculis or Americanum is also called the toothache tree.
Oh.
So, yeah, I think in the past they were used like to chew on the leaves or on the small stems, the ones that are not prickly, to treat toothache. And actually, the sensation that you get from Sichuan pepper is kind of like numbing.
Yeah.
So.
Could be very interesting. And second, just a comment, it's really nice to see, you're using all of the different molecular techniques, but integrating it with all of the other aspects of morphology and chemical and chromosome cytology, because I really think that's just really important for us to all continue to utilize in all of the systematic work.
Yeah. I think especially the chromosome numbers or the ploidy levels is very crucial also for the genetic work. Yeah, so it has been neglected to some degree, but I mean, if something is octaploid and maybe a hybrid or something, then with Sanger sequencing phylogenies, it might come out at a very weird place or something. I think really this chromosome work needs more attention.
Yeah, I mean, there was a time when it was one of the most advanced things that was leading great insights to understanding of different plant groups. And now, hardly anybody does it. So combining the two would be very powerful. And finally, I see that we still have a lot of work to do in Pacific stuff. And Gabe, I think, is going to have some new DNA for us pretty soon.
Yeah. So actually, the Hawaiian project, it was the PhD thesis of Claudia here, but yeah, it's never the end. Yeah, so there's always more to do. Yeah, and I'm looking forward to the new sequences. Yeah.
Another question there. You're muted though, Barbara.
There, thank you. Yeah, I had a question, not so much about citrus, but about DNA sequencing work on herbarium species. And I just wondered, is there anything besides just normal herbarium maintenance that you can do to make it the specimen especially useful for molecular work?
Yeah, I think the most important thing is that you dry the specimen as quickly as possible. And like I think what many people are now doing when they collect herbarium specimens, they also collect something in silica. But if this is not available from herbarium, if it's dried very quickly, then I don't know. My personal record is a specimen from 1861. That was this holotype of this Cuban species, and that worked. But probably it has been collected in the Mediterranean where it was quite dry already. So in the tropics, of course, that's a bigger issue. But in my experience, getting it getting it dry as quickly as possible, that's the most important thing. So I have had specimens from 2020 that didn't work, but things from the 19th century that did work. But of course, it's also largely family or species dependent, and some other things, like I think a colleague of mine works with willows, with salix, and she often has problems. And of course, then secondary compounds may be problematic, but that's in fresh material, but also in herbarium material. But other than getting things dry very quickly, I don't have any other recommendations. I think that's the most important thing.
Thank you.
So I don't see everybody here. So in case someone has a question, just unmute and then start. Because
hi, Mark. Marcos here.
Hi, Marcos.
Hi, thank you for your presentation. It was very nice and interesting. So I have a question. I saw, this wide diversity of fruits across the family, and also leaves. But about the fruits, what do you think are the main evolutionary pressures to, the group is facing? Are they what are the main dispersers? I think some of them are birds, but probably these clades with these giant fruits, the woody ones, like Casimiro, and some other ones probably are big mammals or something. Do you have any insights on the main dispersers?
It's not studied in so much detail. You're right, like the Casimiroa or like some of the citrus relatives are probably mammal dispersed. So what we have very often in Rutaceae are these fruits, the opening fruits, where the seeds remain attached to the fruit, to the pericarp, and those are believed to be bird dispersed. Or it's, for some of them it's known, for others it's proposed, it has been proposed, and I think this has been the most successful strategy. So Melicopi and Xanthoxylum both have these kind of fruits. So one of the sister genera to Melicopi, Euodia, only has, I think, 7 or 8 species, and in this genus the seeds are expelled. So the fruit opens and then the seed jumps out and just falls to the ground. And Euodia has a very narrow distribution area, while Melicopi occurs everywhere from Madagascar to Hawaii. So I think it has been quite understudied, but this strategy with these open fruits, with the seeds remaining attached to the pericarp, that has been the most successful.
Okay. And what about the phyllotaxis? I know some of them, they have opposite leaf and mostly alternate leaf. Do you have any pattern on the phylogeny? The ones with opposite leaf or compound or simple?
We do. So it's like, as always, for some clades, it's very specific. So this Melicopi clade, this rainforest Australian plus New Guinea clade, they are all opposite. I mean, there are 2 Melicopi species with whorled leaves, like groups of 4. but otherwise they are all opposite. The sister group to that is all alternate, and you are right, most Rutaceae have alternate leaves, but otherwise it's kind of scattered. Yeah, so these two clades plus the Auranthioideae, they all have alternate leaves, but otherwise alternate is dominant, but there are opposite leafed genera scattered in there.
I see. Yeah, I know Ravenia with the zygomorphic flowers, they have opposite leaf, and I don't know how many, you know, in the same group, if all of them they have opposite, or is that something like a random happening?
But I think most of the others, so of this Central and South American clade, most of them have alternate leaves, if I'm correct. So I haven't really worked on the South American genera so much, but Ravenia is opposite. Yes.
Yeah. Okay, thank you.
See, there's something in the chat. Oh, okay, just thank you.
Oh. Okay, I'll toss out one more question. You know, going through the whole family, it was really striking that there are so many clades that are made up of a small number of highly diversified groups. I mean, they're mostly monophyletic, it seems, some not, but they're very diversified within these small clades of a few species, and then there's a very large group with good characters. Do you have any thoughts about what are the drivers of these small clades diversifying so rapidly morphologically? Is it different pollinators or dispersal or some kind of ecological differences, or any thoughts?
I just think often the earliest branching clade is always very species poor, and I think these are, and if you look at the distribution, so mainly you have Old World and New World genera there. So I think in these species poor things, I guess there has been a lot of extinction, because many of these genera are monotypic or have 2 or 3 species. So I think this is one thing. And then these outbursts of high speciation, I think, Melicope is one prime example. There we have a classical Hawaiian radiation. But other than that, I think they all had a lot of time to evolve. So it's kind of a large or an old family. Xanthoxylum we have fossils that are 40 million years old. Yeah, and regarding that age, 225 species is not too much, I would say. So the only one where we really have this burst of species, as far as I know, is this Hawaiian radiation of Melicope. Yeah. And it's one of these classical Hawaiian examples. Yeah.
Great, thanks.
I'd like to make a historical comment. I'm old enough. In the end of the last century, a woman who was doing her graduate degree sent a request to the USDA to sample Swingle's Rutaceae collection. She assumed that she was going to be able to get most of the living material of most of the genera out of his living collections. So I was with the Agricultural Research Service in Beltsville. So after about 6 months, I finally found someone. After his death, they kept his collections alive for a few years. And then one winter, they just left them outside and they perished.
Oh my gosh.
Yeah.
And I thought that they would have sent them off to some germplasm repository or something like that. No, they were only interested in the economical ones, not in all these other. He apparently had most genera in the Rutaceae growing in Huntsville at one point in time, but only the agriculturally important ones were preserved.
Yeah, I think Swingle had one of the best collections, maybe the best collection in the world, and it's really a pity what happened to this collection, because I mean, in this book, The Citrus History, he describes what he has in cultivation, or what he had in cultivation, and really these endemic African taxa that just consist of a handful of species. He had all of them, so yeah, he had a great collection, and yeah. I don't know. Yeah, these kind of things happen. It's sad, but it happened. Yeah, would have been a very good resource for my studies. Definitely. Yeah. Okay. One more thing in the chat, but it's also not a question. Okay. Yeah. Any further questions?
Maybe we should let you get a little shut-eye before you have to teach classes tomorrow, whatever you have to do.
Well, classes start next week, so I can sleep.
Oh, okay, great. Anyway, fabulous talk. I really enjoyed it a lot.
Okay, thanks a lot. Thanks a lot. I'm trying to move to the view where I can see everybody, but I don't know. I just do the
There
stop
you
sharing.
go.
Okay. There we are back now. Yeah, okay. So then I hope next time it will be in person. And I can, or maybe I can join one of your meetings in the future. So I hope I can come to DC maybe next year.
Yeah.
And thanks for the invitation. And yeah, so
Well,
have
thank
a
you
good
so
night,
very
everybody.
much.
Thank you.
Yes,
Yeah.
thank you.
Sleep
you.
well.
Okay, thanks. I will.
Thank you.
Okay,
Good
bye-bye.
Bye-bye.
Good night, Marion.
Night, Kathy. Tell David I get dinner and please be in touch with me later this week.
Should have.
That was a good boy. That was a